My name is Habib Rehman and my topic is Biophysics of Muscles and Movement. Biophysics of muscles and movement involves understanding the physical principle that governs how muscle function and how movement is generated in the body. If we need to understand how the muscle function and how what is the mechanism, first we need to know the structure of muscle. So the muscle fibers. Skeletal muscles are made up of long cells in diameter 20 to 100 micrometer, which contains myofibrils, which are further divided into sarcomere. Muscle structure also contains some protein, which help in attachment and which further will lead to the contraction, which are actin and myosin. They are the primary protein involved in muscle contraction. Now I have divided the whole mechanism into 10 steps. This mechanism start with muscle contraction is initiated by a signal from central nervous system. Release of acetylcholine of the neurotransmitter junction, which causes action potential in the muscle fiber and which triggers the release of calcium ions from sarcoplasmic reticulum. Calcium ion combined with troponin and tropomyosin uncovers the binding sites on actin and initiate contraction. Myosin binding sites on actin exposed. Myosin head bind to actin and myosin head executes power stroke, and which actin filament slide towards the center of sarcomere which lead to the contraction. Then again signal from CNS stops. Calcium ion are pumped back into the sarcoplasmic reticulum from where the calcium ion has come. Now the tropomyosin marks the binding site and filament pull back and the muscle and it lead to the relaxation and the actin and myosin will get to its original position. So this is the 10 step where we can understand relaxation and contraction of muscle. Now what are the laws which govern these contraction and relaxation of muscle fiber are force generation mechanism, length tension relationship. Force of a muscle can generate depend on its length. We all know that if a length of a muscle is more, the force which is generated will be more, and maximum overlap which will lead to basically will, if we talk about the mechanism, the maximum overlap of actin and myosin can lead to more, which can generate more force. Motor unit recruitment is the second law. Nervous system recruits motor units to adjust force output based on demand, influencing precisions of movement. This means that if a body demands the movement, that how much movement it should be done, based on the precision of that movement the force is generated. So this is the two factors which on which force generation mechanism depend. Another mechanical property of muscles is viscoelasticity and tension development. Muscle tissue exhibit both elastic and viscous property, allowing it to store and release energy during contraction and stretch. We all know that elasticity is the property of muscle which it inhibit and which can release energy during contraction and stretch. Tension development. Rate of force development can be influenced by the speed of contraction and degree of muscle stretch. We all know that if a muscle is contracted fast, the generation of force will be fast and it will be more. So this state this. Another another law is which govern biophysics law which governs the mechanical movement of muscle is the signal transduction. Basically there are two factors which depend which is neuromuscular junction and action potential. What is neuromuscular junction? The transmission of signal from motor neurons to the muscle fiber involves the release of neurotransmitter. This is called what is called a neurotransmitter junction. Action potential. Muscle fiber generate action potential that propagate along the membrane, triggering the calcium release from sarcoplasmic reticulum. We know that action potential will only be generated when there will be more positive ion in the cell, and the more positive ion cell will come from the inherit of calcium which are present in sarcoplasmic reticulum. So basically what it does, it propagate a signal is propagated through the cell membrane to sarcoplasmic reticulum, and calcium will release into the cell, which will lead to the development of action potential. Now I have come to the conclusion that biophysics tools analyzes the movement of limb movement, which involve kinematics, force involve kinetics, which help us to understand performance in various physical activities. Understanding biophysical principle is essential for application in sports science, rehabilitation, enhancing our knowledge in healthcare. So, if we know the physics which are used in this in biology, so we can understand how we can correct our imagining. This is what is called a sliding filament theory. Now here I am trying to explain you with the diagram how this mechanism goes down. First, here we can say that a cross bridge is formed between myosin and actin and myofilament. Here we can say that it is attached. This is actin filament. This is myosin. It is attached to the actin. Myosin are attached. Now what happens in when there the cross bridge, the myosin perform a power stroke and pulling the myofilament down, it which lead to the movement. So here we can say that the myofilament, the head of myosin provides a stroke which lead to the movement, and here ADP and phosphate in organic is released. Now here what happens when ADP and PI is released, new ATP attached to myosin head and cross bridge detach. Here we can clearly see after a power stroke it has detached, and it has there is separation between the actin and the myosin. Now what happens, the new molecule of ATP comes to this myosin head, and what happens here that it will again come in this erect position where it will in a position to attach with actin filament. So what happens here, myosin uses ATP to return to the erect state. Now what it is in the erect state, now it is ready to join. Now and again cross bridges can be formed with using ADP and PI, and here we can again see that it can get attached to the actin filament, and myosin head get attached to the actin filament, and now again the power stroke will be given, there will be an again movement, then there again will be ADP and PI will be released, then again it come into this position, then again the ATP will come to this myosin head, and then again it will get into this erect position, and this get separated here, and then there will be a condition where it get attached to this actin filament, and then again there will be, and this is a cycle which is called sliding filament theory. This theory is known as sliding filament theory. Thank you.